Let's begin with the marvel at the heart of every green leaf, photosynthesis. This process is the engine that powers nearly all life on Earth, converting sunlight into the chemical energy that fuels the biosphere. Plants, using their unique cellular machinery, capture light and transform it into glucose, the basic building block for growth and energy storage. This transformation is not just a simple reaction, it's a highly orchestrated sequence of molecular events involving specialized pigments and intricate structures within the chloroplasts. As we explore this blueprint, we'll see how each component, from the pigments to the membranes, plays a critical role in sustaining life. Now, let's trace the historical discoveries that revealed the secrets of this process. Journey to Understanding Photosynthesis is a story of scientific curiosity and breakthrough. In the late 18th century, Joseph Priestley discovered that plants could restore air that had been injured by burning candles or breathing animals. Soon after, Ingenhousz showed that sunlight was essential, and only the green parts of plants released oxygen. By the 19th century, Julius von Sachs pinpointed chlorophyll's role, locating it within chloroplasts, the true factories of glucose. Later, T.W. Engelmann mapped the action spectrum, revealing which wavelengths of light drive photosynthesis most efficiently. Finally, Cornelius van Niel's work in the 1930s proved that the oxygen released comes from water, not carbon dioxide. Each discovery added a vital piece to the puzzle, setting the stage for our modern understanding of this life-sustaining process. Now let's step inside the plant cell to see where the magic happens. The chloroplast is a specialized organelle divided into compartments with distinct roles. The membrane system, made up of grana and thylakoids, acts as solar panels, capturing light energy. The stroma, a fluid matrix, is where the assembly line of sugar synthesis takes place. Pigments like chlorophyll, a chlorophyll B, and carotenoids absorb different wavelengths of light, maximizing energy capture and protecting the cell from damage. The light harvesting complexes funnel photon energy to reaction centers, where electrons are excited and set in motion, powering the next stages of photosynthesis. This division of labor ensures efficiency and adaptability, allowing plants to thrive in diverse environments. The heart of the light reactions lies the Z-scheme, an elegant electron transport system. When light excites electrons in photosystem II, water is split, releasing oxygen and protons. These electrons travel through a series of carriers, generating a proton gradient across the thylakoid membrane. This gradient is the driving force behind chemiosmosis, where protons flow back through ATP synthase, catalyzing the production of ATP, the cell's energy currency. Meanwhile, electrons continue to photosystem I, where they help reduce NADP+ to NADPH, a crucial electron carrier. Together, ATP and NADPH power the next phase, the Calvin cycle. This seamless flow of energy and electrons is what makes photosynthesis so efficient and vital for life. ATP and NADPH in hand, plants enter the Calvin cycle, the biosynthetic engine of the stroma. Here, carbon dioxide from the atmosphere is fixed into organic molecules. The enzyme RuBisCO catalyzes the binding of CO2 to ribulose bisphosphate RuBP, forming 3-phosphoglyceric acid 3-PGA. Through a series of steps, ATP and NADPH are used to reduce 3-PGA into triose phosphates, which eventually yield glucose and starch. Cycle must turn 6 times to produce 1 glucose molecule, consuming 18 ATP and 12 NADPH in the process. However, RuBisCO isn't perfect. It sometimes binds oxygen instead of CO2, leading to photorespiration, a wasteful process. Plants have evolved clever adaptations like the C4 pathway to minimize these losses and maximize efficiency. Overcome the inefficiencies of photorespiration, some plants develop the C4 pathway. This adaptation spatially separates the initial CO2 fixation from the Calvin cycle, using specialized anatomy called Krantz anatomy. Here, CO2 is first fixed in mesophyll cells by PEPcase, forming a 4-carbon acid that's shuttled to bundle sheath cells. This creates a high CO2 environment for RuBisCO, virtually eliminating photorespiration. E4 plants thrive in hot, dry environments, while C3 plants are more common in cooler climates. Yet even this engine has limits. Factors like light intensity, CO2 concentration, and temperature can all restrict the rate of photosynthesis. Understanding these constraints helps us appreciate the delicate balance that sustains life on our planet.